Hey everybody, it's Jason again, ASD manager at the Children's Museum of Houston, and we're going to talk a little bit about DNA right now. The activity we did before the DNA activity we're going to do today was about cells, and I talked about how the nucleus of a cell holds the genetic material that when the cell divides is transported over to the other cell. Well, that's through DNA. You might remember we talked about during chemistry something called a polymer. A long chain repeating molecule. Well, DNA is that. DNA is actually really big. I know cells are small, you have to look at them through a microscope, and I know that the DNA must be smaller because it's in the smaller unit of the cell. But if you actually took DNA out of the cell, the nucleus of a cell, and you stretched it out, it would be 2 meters long, 6.5 feet long, one strand. That's pretty amazing. So DNA is basically has this sugar phosphate backbone. And connected to that sugar phosphate backbone are nucleotides, nucleotide bases. And there's 4 of them. There's A, adenine, there's C, cytosine, there's T, thymine, and there's G, guanine. C and G hook up together in any kind of combination. G can be first, C can be first, A and T hook up together in any kind of combination. And those hookups are through nitrogen bonds. So when you look at DNA, it actually looks like a stepladder when it's unfolded. But in order to be folded into the nucleus of a cell, even though it's 6.5 feet long, these proteins called histones allow it to wrap itself into something called a double helical strand and compact itself into that DNA or into that nucleus. When a nucleus is dividing, that DNA unfolds and transports its genetic material into the new nucleus of the new cell. What we're going to do right now is we're going to try to extract DNA from wheat germ. This stuff right here. Now, if you recall from the botany lessons, wheat germ is a plant. So we're going to actually extract DNA from a plant and see if we can actually pull enough of it out to put inside a little container that we can take home with us. You guys want to give this a shot? Let's do it. First thing we're going to need is a conical tube. This little guy right here. Now normally with this conical tube you'd also want a conical tube rack. Unfortunately we don't have the rack. Okay? So I'm using a cup as a substitute to hold my tube because I'm doing this all by myself. If you have a friend with you, you can use your friend to hold it. We're going to uncap the tube and we're going to go ahead and put about 2.5 milliliters of wheat germ inside the tube. 2.5 milliliters would be like right about here, where it ceases to be a cone and starts to be the cylinder, okay? So we're gonna go ahead and put that in. You don't need much to extract DNA. So see, that little spoonful is about all you need, all right? And don't worry about if stuff sticks to it or whatnot, that's okay. I'm gonna put the cap back on and get ready for the next step, which is we're gonna add water. So I'm just gonna put water in this cup right here, so that it's easy to pour into this cup. And what we want to do is we want to get to the 20 milliliter. All right? We don't need that much water, we just want to get to the 20 milliliter. So we're going to very carefully pour water inside until we get to the 20 milliliter, which is right there. Okay? I'm going to put the cap back on just for a moment to get my next tool that I will need. My next tool is a skewer. What I'm going to do with this skewer right now is I'm going to smash the wheat germ with it. The reason why I'm going to do that is, if you recall, plants have cell walls that are protective and rigid. We got to break through those cell walls so we can get into the plant cell, the organelles, the nucleus, so we can get that DNA out. So we're going to go ahead and smash up some plants, some wheat germ. you want to use the flat end of the skewer. We're going to use the pointy end later on for something different. You want to do this for a good couple of minutes, okay? So you might want to switch hands every once in a while. If you have a partner, you might want to use a partner. So there we go. Smashed it up. Looks a little cloudy. That's good. We're going to go ahead and cap it up again. And we're going to very gently shake it up. We're going to gently shake it up. All right, and we're going to set it aside for a second. Let it settle. Next thing we want to do is we want to make a soap solution. Soap is a surfactant. It draws fats to it and grease so it can separate it off other things. What we want to do with our surfactant in this case is we want to draw the lipids and the proteins away from the DNA so the DNA has a chance to precipitate out into another solution we're going to do in a little bit. So we want to fill up a cup with water and we want to squirt a little bit of soap in there. Let it mix up. You can actually use your skewer to mix it up. Then we want to let it settle for a second because we don't want to get too many soap bubbles inside of here. You can get a little bit any time you try this, but you just don't want to get too many. So what we're going to do now, we're going to uncap it again. We're going to use our pipette. We're going to fill it to the top, which is about 3 milliliters, not 30. Just by squeezing it and filling it up. And then we're going to very gently squeeze it on top of our water. That'll reduce bubbles. And we're going to do a second one, okay? Again, very gently. Don't want to rush. So, once we have that soap solution in there, what you want to do is something called whorling. You want to take it in your fingers like this and very gently whirl it around. You want to get those fats and those lipids from the wheat germ onto the soap so that we can get the other stuff out that we want to get out, which is the DNA. So we're going to do this for a moment. Again, don't go fast. Don't worry too much if you have soap bubbles. You can always fix that later. What you don't ever want to do though, if you have soap bubbles, is poke it. Just let it go. You want to whirl it for a good couple of minutes. Then we'll have our final step. Okay. Our final step is to use alcohol to allow it to have a place to precipitate into the DNA. The alcohol will be less dense than the soap solution, so it will rest on top. The DNA will be less dense and float up into the alcohol. It's going to look like snot or like spider webs, so it's kind of gross looking, but it's pretty cool when it happens. This is 91% isopropyl, so it's easily found in stores. So you want to use a fresh pipette, not one you used before, and for the soap solution. And again, fill it up. This time though, tilt it and gently squeeze it in so that it stays on top. And again, do two. And you can see the layer right there. And already I can see DNA climbing into the alcohol layer. It's that snotty looking stuff. So what you can see floating up into the alcohol, that sort of snotty, slimy looking stuff that looks like tendrils, like little straws coming up, that's your DNA. What we're going to do is we're going to take that DNA and put it into a little container so you can take it home with you. I'll show you how to do that right now. So here's what we're going to do. This is called a microcentrifuge tube. All right, this little tube right here. I'm going to use the sharp end of my skewer And I'm going to twirl it around the DNA like spaghetti. I'm going to grab it, twirl it around. And I got me a nice big piece. You can see that coming off right there. And what I'm going to do next is I'm going to stick it inside of here and attempt to get it off. Now that's the hard part sometimes, is getting it off the skewer once it's in there, or once it's on there. So you just got to be patient. You can also use a Q-tip to try to get it off and spread it around on there. And I'll try to get some more right now. Ooh, that was a good piece. There we go. There we go. That's a nice piece of DNA. You can see it right there. That's a good piece. And what I'm going to do next is I'm just going to put some alcohol in here to preserve it. And you can see it's going to float down to the bottom. That's a nice little piece of DNA right there. You can see it floats down the bottom. It's a nice little piece of DNA right there. Now, I'm going to close it for a second, but what you can do is you can make yourself a little necklace. Just take a piece of yarn, cut it off, open the top, put it around the cap, close the cap, and then you can wrap it around, you can put it around your neck, and that's how you can take it home. Just like this. And you can say to your parents, hey, look, I have some DNA from a plant. And that's actually what it looks like.